Inside a neonatal intensive care unit, a newborn presents with progressive lethargy, low muscle tone, and rapid breathing. Sepsis is the initial clinical suspicion, though the patient is actually experiencing a total metabolic collapse. The true diagnosis reveals itself when the blood work returns from the lab, displaying a critical plasma alanine value exceeding 1,500 micromoles per liter. That single number is 3 times the normal biological limit. Understanding why that specific molecule triggers a hospital alarm requires looking at its evolutionary history. Alanine is one of the original, primitive amino acids. It was selected by the chemical environment before complex life even existed, and is frequently found preserved inside ancient prebiotic matrices like meteorites. The ancient, simple structure of this molecule decodes the neonate's systemic failure. Alanine's entire functional utility comes from its remarkably compact architecture. It is an aliphatic, non-polar molecule, possessing the absolute smallest side chain of all hydrophobic amino acids. That side chain is just a single, uncharged methyl group. That lack of bulk gives it almost zero steric hindrance. Instead of acting like a massive grinding gear, it acts like a microscopic ball bearing. It slips into the core of complex protein alpha helices, allowing them to fold tightly inward without their chemical parts colliding. Human biology depends on precise molecular handedness. Human genetics and ribosomal machinery exclusively read the L enantiomer configuration to build tissue. We hoard this specific material. Roughly 70. When you push your skeletal muscles through an intense sprint or a prolonged fast, those cells burn energy rapidly and eventually break down internal proteins to keep moving. This catabolic process strips away amino groups, generating raw ammonia. Ammonia is violently toxic to human tissue, and skeletal muscle lacks the enzymatic machinery to neutralize it. To prevent hyperammonaminic shock, the muscle relies on an enzyme called alanine aminotransferase, or ALT. The muscle cell uses ALT to rip the toxic nitrogen off the ammonia and forcefully attaches it to a molecule of pyruvate, a natural byproduct of burning glucose. The instant that binding occurs, the toxic waste becomes stable, harmless L-alanine. The thermodynamics of this chemical reaction are highly unusual. The transamination of pyruvate to alanine has a standard free energy change of near zero. Because it is dictated entirely by concentration gradients rather than direct cellular energy, it operates like a frictionless pendulum, costing the body almost no ATP to execute. Once the toxic nitrogen is safely packaged into alanine, it is pumped out of the muscle cell and cruises through the vascular blood compartment until it reaches the liver. Liver hepatocytes extract the alanine and use that exact same ALT enzyme to run the chemical reaction in reverse. They separate the toxic nitrogen to be safely excreted as urea, and they take the leftover pyruvate carbon skeleton and convert it back into fresh glucose to feed the exact muscle that sent the waste. Alanine operates as an infinitely recyclable biological transport vehicle. It shuttles lethal waste across the body in a closed loop, ensuring the organism can exert extreme physical effort without poisoning itself. Because this entire shuttle system relies on flawless internal routing, any roadblock causes immediate downstream chaos. In a genetic crisis state known as pyruvate dehydrogenase, or PDH deficiency, the primary intake valve of the cellular mitochondria ceases to function. Under normal conditions, pyruvate enters the mitochondria to be burned for massive amounts of energy. With a defective PDH valve, that pyruvate cannot cross the threshold. It begins violently backing up and overflowing the cytoplasm of the cell, threatening to halt cellular respiration entirely. The cell enters a panic response. To clear the factory floor and prevent immediate metabolic death, it desperately converts this massive backlog of unburned pyruvate into lactic acid and alanine, dumping both into the bloodstream. This sudden surge of alanine flooding the plasma serves as the chemical evidence of a massive metabolic traffic jam. The spike serves as an early warning alarm bell. It alerts the clinical team that the patient's mitochondrial engine is starving, giving them a narrow window to intervene before total systemic failure sets in. Tracking this specific traffic jam requires extreme laboratory precision, and clinicians must separate the measurement of the ALT enzyme from the measurement of the free alanine substrate. A massive spike in the free alanine substrate indicates the mitochondrial blockage we just mapped. Extreme levels of the ALT enzyme floating in the blood indicate acute hepatocellular necrosis. The structural membranes of the liver cells are actively rupturing and spilling their internal enzymes into the vascular system. To find the mitochondrial block, the lab must accurately measure the substrate itself amidst a fluid containing thousands of other interacting chemicals. They rely on liquid chromatography tandem mass spectrometry, or LC-MSMS. The triple-quadruple system acts as a highly specific sieve, filtering out everything except molecules weighing exactly 90.1. To prove identity, this isolated molecule enters a collision cell, where argon gas physically shatters it into a distinct 44.1 fragment, providing undeniable quantum proof of alanine. This extreme level of molecular ballistics guarantees specificity. It is the only definitive way to track invisible metabolic traffic occurring deep inside human cells. Despite multi-million-dollar physics, a completely mundane human error at the bedside can ruin the data. Human red blood cells carry massive hidden reservoirs of intracellular alanine. If too much vacuum pressure is used, mechanical stress tears the blood cells open, leaking their internal payload directly into the sample. This intracellular hemolysis escape creates a fake metabolic crisis on paper. To defend against this, hospital computers deploy automated delta checks, halting the release of any results that shift by impossible rapid margins until a technician can physically inspect the vial for popped red blood cells. The human baseline is further complicated by the trillions of passengers living inside the gut microbiome. Certain commensal bacteria actively synthesize a mirror image D-alanine isomer to build their cell walls, and this alien molecule continuously seeps into human circulation. Because human tissue strictly requires the L configuration, the liver is forced to constantly deploy specialized D-amino acid oxidase enzymes to hunt down and clear this bacterial chatter, protecting the integrity of our native signaling network from microbiome interference. Back in the ICU, the neonate's true baseline is established. The confirmed PDH deficiency means their mitochondria cannot burn carbohydrates. The treatment involves immediately eliminating those carbs and implementing a strict high-fat ketogenic diet. Fat metabolism requires mitochondria, but it utilizes a completely different cellular doorway. The liver converts the dietary fats into ketone bodies. These ketones bypass the broken PDH bottleneck entirely, entering the mitochondrial cycle downstream to safely restore energy to the starving brain. By measuring the exact flow of this single simple amino acid, clinicians map the hidden intersections of cellular machinery. That data provides the architectural blueprint required to bypass genetic blockages and pull a patient back from metabolic collapse.